It's our pleasure to have Professor Areejit Samal today to give us the talk. Professor Areejit Samal obtained his PhD in physics from University of Delhi. He has been a postdoctoral fellow and visiting scientist at multiple international centers like International Center for Theoretical Science, Trieste, University of California, Berkeley, Institute for Systems Biology, Seattle, University of Paris, Zug, 11, Orsay, Max Planck Institute for Mathematics in the Sciences, Leipzig. Currently, he is the head of the Computational Biology Group, Institute of Mathematical Sciences, Chennai. He will talk about linking exposome and health by leveraging data science. Prof Samal.
Thanks, Kalyan. Let me just start by thanking Kalyan for this kind invitation to Krea. It's my first time here, and I've been enjoying the visit, interacting with colleagues here. What I intend to do is I'll tell you about an area where we have been working on for the last 6 years, and it has sort of crystallized into a theme around which computational approaches can be applied and a lot of different questions can be answered. I think in this audience now, I think all of you are aware of IMSc, or so just to review a brief overview of IMSc. IMSc has been there for now more than 61 years. It was established in 1962. It started out as an institute of basic theoretical research in physics actually. Then subsequently there was a group in mathematics which came about, and subsequently theoretical computer science. And since 2013, a new group called computational biology. So these are the four main areas of IMSc. We currently have roughly about 50 faculty members. Our center strength is 16. We only train PhD students and postdocs. We have no undergrads, so it's a purely research institute. We are an autonomous body of DEDuCT, so we are well funded. And our degree comes from the umbrella institution, Homi Bhabha National Institute, which is a degree university, which grants PhD and other degrees, various autonomous bodies at country other than India. And of course, if you have not been to IMSc, it is basically the heart of the education and IT hub of Chennai. We are surrounded by IIT Madras, Anna University, CLRI, and you have across this OMR going down for 20 kilometers, we have software. We have also biotech parks and so on. So we are actually very well placed to be in the academic, industrial, software ecosystem. So before I go on and present the piece of work that I want to talk about today, over the last 9 years since I have been in IMSc, I started my different career. These are broadly the directions I have been working on, and these are some of the representative publications I have to show what we have been largely doing. It's a bit spread out, to be honest, but what I will be talking today basically lies in this first area, which is where we are looking at chemical spaces, and chemical spaces which are relevant from 3 different perspectives. One, drug discovery, which I won't be talking about today, and second, systems toxicology, human exposure. That is what I will talk about. Of course, my core training is in network science, and we keep continuing to develop methods for the analysis of complex networks. In particular, in the last 5 to 6 years, there are two directions here we have been taking. One, within the sort of the mandate of this mathematical partner group that I had, which is to sort of develop geometry-inspired measures for edge-centered analysis of complex networks. But essentially, we have quoted concepts from discrete geometry, especially this Ritchie curvature for cone Ritchie curvature, to the domain of complex networks, and shown its utility in analysis of real-world networks. In particular, we have analyzed networks in finance, analyzed networks in neuroscience, and so on. Another area we work on here is to develop tools for modeling field-regulated networks, their reconstruction, and dynamics. And in this, there are these two pieces of work which have recently come up, which I alert you if you're interested in what sort of modeling work we do in our lab. Lastly, I also have deep interest used in modeling metabolism in patient system. This was actually funded by a American fellowship. These are some of these things that we have done since I have joined. For example, we have a pipeline to predict the secreted proteins in microbes, in particular fungi. And recently we have come up with a tool which can predict a certain type of effectors which are secreted by titin secretion systems in the bacteria, in a phylum of bacteria known as Bacteroides. These are things where we take a lead with my students, and we of course show our utility by collaborating with experimental biologists. And I would say here we play an enabling role where we essentially help various experimental groups in the country and abroad who generate a lot of data, typically high throughput data of omics nature. We try to analyze them within network context and give a better understanding to the biological processes they underline them. So that's broadly the 4 areas we work on. I must say that in the last few years, more and more, these are the 2 areas we heavily work on, and in particular the first area. So, and my talk will be a sub part of the first area, and the first area can be further subdivided into 2 kinds of chemical spaces that we study. And this sort of I present as compilation, curation, computational exploration of chemical spaces. And there are 2 types of chemical spaces we have been working on in the last 6 to 7 years. One which are natural product spaces, and this space is potentially beneficial to humankind. And second, environmental chemical spaces which are potentially harmful to humankind. I won't talk at all about this work, which has gained a lot of attention. In particular, we work on phytochemicals of Indian medicinal plants, cytochemical medicinal fungi. I won't touch upon at all this work. What I want to focus on today is the second type of chemical spaces, where you have chemicals which are in the environment, which we use in our daily environment, or come across in our daily environment, and they could be potentially harmful to us. And there again, we have worked on various chemical spaces, and I will particularly focus on one particular type of toxic chemicals which are known as endocrine disruptors. So all of these have led to resources which are in the square brackets that you see, which compile all the curated data that we have gathered and plays an enabling role for researchers working in these different areas, and these are openly accessible to everybody to browse, and even there are platforms to create and so on. Yeah.
I think so. You talk about so there's not kind of the mode of contact does not matter, right? I mean, is that what I'm writing, or is that basically what you're talking
You are jumping too far. The mode of contact matters, the dosage matters.
So it can be basically there are like basically something we ingest as well as something that is present in the environment. Both of them. Sorry, is that? I mean, in the sense that we classify the environment in the spaces.
So here the classification is large. So there can be multiple classification. The first, and these are not exclusive of each other. There is overlap among the same chemical can be neurotoxicant and endocrine disruptor, and also be in the milk exposome. So let me just dive in and explain shortly what we are trying to do. So we are on the right track. Yours question.
No, I think part of the question came with the discussion. I was wondering whether all these potentially harmful chemicals have a level of toxicity, which they are not. Is that factored into this whole?
Yeah, so I will come to it. So we particularly have the first resource, which is the only resource of that type which captures that kind of information. There is no resource in the world which has a comprehensive information of that kind. That is why it is called information. So there is a term in my title which is exposome, and most of you may not have come across this term called exposome. What is exposome? The exposome essentially encompasses the environmental exposures, including the lifestyle factors which individual encounters from the prenatal period, that is, from the time it is in the mother's womb and so on. It's a very complex thing to characterize. Of course, an individual's exposome varies depending on the environment where you work, live, and play. And although in this talk I will talk about the specific type of exposure, which is chemical exposure, but the exposome not only comprises the chemical exposure, but also things like stress, radiation, microbes that we are exposed to. So the concept of exposure is an overarching umbrella, term which covers all of these exposures. But I will particularly focus on the chemical part of it. Of course, the completion of the Human Genome Project, and the outcome was a landmark event. And certainly, the Human Genome Project was touted as the blueprint to life. That is, with the sequencing of the human genome, we would understand the key to various diseases and we'll come up with therapies for each of the diseases. But if you see today, we are largely facing, or a large fraction of the population is facing a lot of complex disorders or complex diseases like diabetes. And if you try to see how much of these diseases, complex diseases, you can explain by purely genomic variation, you can only give a partial explanation. And perhaps the other part of the explanation lies in the environmental factors. So I'm trying to oversimplify it for the audience. But to put simply, one should not think of health as a function of only genome, but rather health is a function of both genome and exposome. So, like the Human Genome Project, there is a need to characterize the exposome, all the exposures that we are encountering, to understand the environmental factors that are affecting it. And in this line, in the last 5 or 6 years, there is a push to also start a Human Exposome Project, which has been already launched in Europe. So, of course, having made the case for characterizing the exposome, I would like to make a case for computational and in particular data science or network science approaches in their utility in this area. So, as I mentioned, human exposure to environmental chemicals is an important contributor to the disease burden. And the exposome not only consists of the chemicals that we are exposed to, but also things like radiation, stress, and so on. So what has happened is that I will make a case for why computational tools are further needed in the area of computational toxicology tools next down the line. One of the things which should be noted is that this is a perspective in science which has come out in early 2020, which is authored by two of the pioneering researchers, Vermeulen and Miller, in exposome research, and also one of the most pioneering researchers in network science, Laszlo Vargas. And what they call for is a need to utilize tools from network science to link exposome and health, which basically is at the interface of chemistry and biology. And in particular, what we are looking at is to trying to link the chemical exposure leading to some perturbations in proteins or genes, and that being reflected at the level of the interactome, explaining the adverse effects at the large scale. So basically, one of the goals that we are pursuing in our group is basically to both enable and advance the link between the chemical exposome and human health using computational approaches. Actually, we are using very simplistic approaches and able to get very interesting results already. But there will be a lot of scope for mathematical modeling, predictive modeling in this area, and I will give you a reason for that, why it will be sought after. It's already sought after. It will be sought after. So before I go on to the particular piece of work, so this is that the work has been what we have achieved. These are 7 papers which these 2 people wrote, and the work was really led by my PhD student who graduated last year, January. So and the postdoc we started. So we have characterized these different exposomes. So we have characterized endocrine disrupting chemicals. First is source of neurotoxicants. These are chemicals which have been detected in actually breast milk. That is, they somehow reach the breast milk from the mother. And I won't cover there, but we show that there is enough evidence that these chemicals could actually affect both the lactation pathways and immune signaling pathways. This is a resource for fragrance chemicals which are used in children's products like toys. You may see that some toys smell very good, but is it actually not toxic to the child or not? One needs to characterize. So I won't touch upon any of these resources, but we'll only talk about the endocrine disruptors. So essentially, these are extremely valuable resources which society at large can use to get themselves aware of what can potentially happen. And of course, they are also enabling the research areas of human exposome and computational systems. So I'll focus on endocrine disrupting chemicals. So what are these endocrine disrupting chemicals, or for short EDCs? So according to the World Health Organization, the definition of the endocrine disrupting chemical is as follows. An endocrine disruptor is an exogenous substance or a mixture that alters the functions of the endocrine system and consequently causes adverse effects in an intact organism or its progeny or subpopulations. And they of course consider such chemicals as chemicals of emerging concern. These EDCs are actually omnipresent in our environment, including air, soil, and water. Few of them are actually come from natural sources. They are in plant-based foods. Of course, at certain dosages, they become endocrine disruptors. While most of the EDCs are synthetic chemicals, which chemists have produced for the benefit of the humankind for various purposes. For example, in pesticides, cosmetics. There is a lot of awareness and advocacy going on surrounding these particular class of chemicals in the West. In particular, here's a document or a non-binding resolution which the European Parliament passed in 2019, calling for a comprehensive European-wide framework for endocrine disruptors. And within the Europe, even France is taking lead in framing a national policy to limit such chemicals in use. So, looking ahead a bit about the biological mode of action by which these chemicals disrupt the endocrine system. There are broadly at least two different ways by which these chemicals can disrupt our endocrine system. So the endocrine system essentially consists of a bunch of glands in our body which secrete different hormones. So essentially the chemicals, if they enter our body and start mimicking the natural hormones, then they compete with the natural hormones to bind with the receptors. And sometimes you don't expect the hormone to be there, but the EDC mimics the hormone. It binds to the receptor and leads to a cellular response, but it's not expected to be there. Also, they can disrupt the endocrine system indirectly by changing the hormonal levels in our body by affecting the hormone metabolism. Right? That's not very important to understand, the talk which I am doing today. But this is a very complex, network of glands and hormones, and these are at least two different ways by which the chemicals could disrupt the endocrine system. So, in short, if the chemicals disrupt the hormones produced by these different glands in the body, you can have from developmental disorders to fertility problems. Okay. So, they are a cause of concern. So, before I move on,
Just one question.
Yeah.
Is that the binding of these chemicals
Yes.
onto those sites? Is there a proposed mechanism or has it been demonstrated to be?
For a few chemicals, it can.
for others, there are computations. But for most chemicals, the actual mode of action still needs to be read out. So I'll come to that part also, how we are trying to help advance that also. So I mentioned this area of exposome and human exposome. There is another perspective to it, which is the new emerging field known as computational toxicology, computational resistance toxicology. And what this field tries to do, let's try to understand. So toxicology is a traditional field, which is a sister science of pharmacology. It basically studies the adverse effects of chemicals on living organisms. So the traditional method of characterizing the toxicity is basically animal testing of chemicals. But there is ethical concern to that because there is a need to replace the animal testing with alternate methods because we don't want to torture the animals for testing. Now, moving on, if we look at just the United States and just the documented number of chemicals which are used in various daily life products, the number of different chemicals is at least 100,000 and much more actually. This is just in the United States, what is documented. But if you look at how many of these chemicals which we come across through the daily use of different products have been tested for toxicity to any extent. And the most comprehensive testing has been done by the US Environmental Protection Agency or the US EPA. It has tested roughly 10,000 chemicals. So even if we believe that there are only 100,000 chemicals which are in use, 90,000 of them have never been tested for safety concerns. Now, given the ethical issues with animal testing, it is, and also the costs involved, it is perhaps infeasible to do all of that. And as you know, chemists are very prolific at producing more and more chemicals with newer and better properties. So we need to keep emergence of new chemicals which are being produced by chemists on a daily basis. So computational toxicology is basically an emerging area which aims to build mathematical models to predict the adverse effects of chemicals on living organisms. I have given a justification of that. If we have very good predictive computation models, we can quickly predict for the remaining 90,000 chemicals if they are going to be toxic. So one of our long-term goals, which we have certainly not achieved so far, is to build network-based predictive models for toxic chemicals, in particular endocrine disrupting chemicals. And certainly, in order to build predictive mathematical or computation models for EDCs, there is a need to create highly curated data sets for EDCs. Why is that? Because you're trying to build a predictive model using data which is trash, then you know what your results are like. So you need a highly curated data set, and we started actually working in this area with this approach. So what we felt is that we first, working up for 6 months, trying to build some network-based models, we realized that none of the data sets which exist today when we start this work serve the purpose. So we felt that there are certain features we desire with a, if I say so, a training data set, which can ideally serve as a good, data set to build models. And the properties that we desire were as follows. That is, first, the data set should have chemicals which are supporting evidence in humans or rodents. What happened is that when you look at the resources which existed till then, on EDCs, endocrine disruptors, they compile information not only from humans but computational studies, fish studies, environmental monitoring studies, rather than exclusive focus on EDCs, with experimental focus on humans and evidence in humans and rodents. Secondly, the endocrine system, spread across the body. One needs to also compile the adverse effects. One chemical may cause fertility problems, another could cause developmental disorders. They may not cause both of them. So we wanted to also, and these previous efforts which had compiled EDCs did not compile experimental evidence for which are the particular adverse effects which they cause, specific to humans. So that's something we want to catalog. Number 3, which was asked by 2 of you, we also need to compile the dosage of levels of EDC, because even water can be toxic at some dosage. So, and in particular, we wanted to capture the dosages at which different adverse effects are observed. Because you may observe fertility problems at a certain dosage, while developmental disorders at other dosages. This is a very complex problem, but we need as much data as possible to be much better predictive. And with these goals, we designed a 4-stage workflow to identify EDCs with supporting evidence in humans and rodents from published literature. So we didn't, of course, do any experiments. We went back and tried to mine the literature, which was vast by then, to see if we could create a resource of this type. So I won't go through the work of 2 years of compiling the data. I don't know how my student agreed to do this, but let's go over what we managed. So first we did a sort of a keyword search and captured articles which were there in existing resources, which are likely to contain experiment evidence or EDCs. And we roughly got about 14,300 studies. Okay? And essentially for these 14,300 studies, abstracts had to be read and filtered out for potentially information on EDCs with experimental evidence in humans or rodents. And subsequently, we looked at full text and we wanted to really only capture evidence which is in vivo or in vitro experiments in humans or rodents, and we filtered down to 3,300 studies. So this 3,300 studies has some information. Now for these 3,300 studies, we had to get the full text, and we had to look at which chemicals were tested in each of these studies, because one chemical could be tested in 15 studies or 15 chemicals could be tested in one study. So we created a mapping of chemicals to studies, and there were actually 1,626 unique chemicals. There was a lot of work, which were covered in at least one of the 3,300 studies. If that seemed a lot of work, hang on, so some work, but the next stage was even more important. So once we had this 1626 to 3,300 study mapping, we we went ahead and did this stage 4, which is a subjective assessment of the actual experiment reported in that paper to decide if we can call a chemical to be a potential ADC. Okay. We always say it's a potential ADC because we make a subjective decision based on some experiment published errors. So here I want to mention that this workflow I wanted to go through quickly was inspired by actually a consistent statement or a framework which was laid down by a leading scientist in the area who met in actually the German Federal Institute in 2016 and published a consistent statement for identification of ADCs. And this actually guided our 4-stage workflow which we have designed. In particular, I want to emphasize that they wanted the identification of a chemical as an endocrine disruptor to rely on the weight of evidence evaluations of both the adverse effect and the mode of action. And these are aspects which no other resource prior to our work had ever taken, other than the 3 features which are there in our data set. So with that, I won't go into the details. The students actually read, one student read every paper and assessed if we could rely on the experiment to call it as potential. And finally, what we were confident in was to call 686 chemicals as potential ADCs. Whose support came from 1796 of the studies of the 3,300 studies. So once we had this final list of 686 potential ADCs, we compiled further information which allow computations to be done. Its chemical structure, what is the source in the environment, chemical classification, and a bunch of other things.
So how do you encode the chemical structure?
It's very standard. To give a 2D structure, there are several formats in which it can be decoded. One is STF format. You can actually download all the structures from our website in different formats.
Well, you mentioned structure. We know in chemistry that isomorphism, requiring structures, one of them can be toxic and the other one not.
So we resolve the stereochemistry. We try to resolve the stereochemistry as much as possible. I'll show you some examples of that, of how very little change can lead to very large change. So I'm summarizing a work of 3.5 years in some slides. Okay. So now, what we had, we compiled, of course, the data, and and we published a resource called DEDuCT. Okay, it is copyrighted to the institute. When we released version 1 in 2019, it covered the 686 CDCs with all those other additional features and evidence from 1796 articles. It caught the imagination of several people and stakeholders. So we actually did a different type of study, which I'll passively mention with a different perspective, which is very interesting. In 2021, we updated that. And we now have 792 potential ADCs with evidence from 2,200 articles. So there is a need to keep it up to date, and we'll get to it why later on. But the thing is that DEDuCT is clearly a resource which is very useful. But I have friends like Prof IMSc, and they will say, okay, he's just doing stamp collecting. He's not intellectual enough to be in IMSc, right? So, very important. So then you have to show the utility of the resource, what you can learn from it. Before we go on, let's address another elephant in the room. That why did my student read through all the papers, and can maybe ChatGPT do all this? Okay, in an NLP fashion. So this is one of the 1796 papers, and perhaps the easiest paper, which was providing evidence. So I have actually annotated PDFs of this type for each of the 1796, actually 2,200 papers, which give the evidence. So if you see, this paper talks of a chemical whose name has been picked up by my student, the type of study that was done in mice, the kind of test concentrations which was there. Then she read this and understood that the adverse effects could be actually what they were reporting is DNA damage and apoptosis in ovarian cells and affects follicular. So this is a controlled vocabulary we create, because this is not always exactly this. So people, the same thing could be reported in a synonymous fashion. We map it to a sort of a controlled vocabulary for adverse effect. And finally, we also classify these adverse effects into 7 categories, and this was classified as reproductive effect. Okay. And finally, which dosage at which first time the adverse effect was observed, and that's this 20 microgram per kg. Okay. And then you can actually get a graph like this. Okay. So you can convince yourself that ChatGPT cannot do it. Okay, so let's try to see what we can learn from this data. So we did rudimentary analysis in the first paper, which I am important, and then an extensive analysis of the sadness and subsequent data. So US EPA actually releases a list of chemicals which are considered to be safe chemicals. That is, if you are in the safe list, then if you use it in your product, you are not regulated. So the first thing we found is that when we did the analysis, a comparison of the 686 EDCs in our list potential and their safe list in 2019, we found 10 chemicals to be potentially disease. Further, there is also another list by the Food and Drug Administration of list of inactive ingredients which are allowed in different drugs. For example, you have seen that the capsule may have a color or may have a protein. That is an inactive ingredient. The active ingredient is inside it. So there are a bunch of inactive ingredients which are allowed. When we analyzed the inactive ingredient list, 44 chemicals which are approved by FDA at that time were actually potentially diseases. What I can tell you very nicely is that after 6 months when we analyzed, some of the ones we had found to be potentially diseases were removed as allowed ingredients by FDA. Finally, okay, so these chemicals are not just here, but if you look at the environmental source information in our resource, they are found in cosmetics like shampoos, soaps, hair dyes, household products like dishwashing liquid, food, emulsifier, and so on, pharmaceutical products like eye drops, diaper creams, and so on. Further, to give support that we have actually the chemicals which are serious in nature, there is a list of 1328 chemicals according to EU regulation which are banned in cosmetics, and 101 of them are actually not 686. So this clearly shows that we have things which are really toxic in our list. Okay, so I'll present 2 slides of the kind of network analysis one can do with this data set. So we have 686 EDCs, and one question one can ask is that how similar are 2 chemicals to each other? So you have 2 EDCs and you have a metric for chemical similarity. This is a distance metric between 0 and 1. Let's not go into the details. There are standard procedures to do it. So I will put an edge between 2 chemicals if this distance metric, which is Tanimoto coefficient, is greater than 0.5. Okay. And I then view the whole list of 686 EDCs as a graph, as a network, where each node is one of the EDC, and there is an edge between 2 chemicals if they are sufficiently chemically similar. Okay. Now, if you look at it, there is a large connected cluster of chemicals which are EDCs, and they can be further partitioned into modules using a modularity algorithm, which is Lua modularity. But there are many disconnected clusters, and there are also isolated chemicals which are not similar to any other chemical. This suggests that the space is quite chemically diverse. This suggests. What is interesting is that the color of each of the chemical here is based on the number of benzene rings which are there. As you can see, modules typically consist of chemicals with similar number of benzene rings. And this was a blind approach, but is trying to get this back very simply.
Benzene has a structure.
So you can just count how many benzene rings are there in each chemical. So and the number of benzene rings is so it's green means 2 benzene rings.
All of them have some benzene.
Not always, no benzene.
Oh, no benzene.
So that. But then you can also see some interesting things. You look at this 2, there are 1 benzene ring, and this cluster 11 there is also 1 benzene ring, but they are not clustered together. And then you see one something, it's a chlorobenzene ring, this is a bromobenzene ring. You can see this kind of a chemical similarity network analysis can actually in a blind fashion reveal many insights on it. I won't go into depth of what other things you can learn from here, but that's one type of analysis we do. So that is a similarity network of chemicals, and this is the approach we take to all our chemical spaces in particular.
Just for a detail,
Yes.
you have naphthalene, you have two benzene rings.
Yeah.
You have biphenyl, which also has two benzene rings. So will you just count them both as being equivalent? Or will they be
I think this is agnostic. Actually, one of my students was an expert on chemical-informatics. That's why I could do it. I have very little knowledge of chemistry. I can say it. So, second type of network that we constructed was I didn't tell you this, but for the EDCs that we have, the 686 EDCs in our initial list, we could get from some of the experiments which are the genes or proteins which are the direct targets of it or indirect targets. So you have a map of chemical to gene, okay? And from this bipartite network, you can actually create a chemical network based on what is the similarity of the target genes between the two chemicals. So you can basically use the Jaccard matrix, which is the cardinality of the intersection between two sets by union, to define, to figure out what fraction of genes targets between two EDCs are shared. And you can take cutoff, it takes a cutoff to be high enough, and again make a EDC network based on not target similarity, not based on chemical similarity. And this data I must mention was not available for all 686 EDCs and was also partially available. It was available for about roughly 380 odd of our chemicals. Again, what you see that there is a large connected component, many isolated EDCs, and some smaller clusters. Here, the node size has been drawn proportional to the number of links it has to other chemicals in the EDC. So, if you see these two chemicals whose PubMed IDs, PubChem IDs I have mentioned, this is actually the pesticide DDT, and this is well-known carcinogen of telecom. So you can also take a target similarity network and get some insights from it. This is another type of analysis which I present for EDCs, but we move for other chemical spaces as well. Now, one question which is in your mind is that how correlated are target similarity to chemical similarity? So for the chemicals which we have the target information. For those chemicals which can pairs for each pair of chemicals, we can plot the target similarity to chemical similarity, and if you look at the correlation, it is really weak. Now, this has an implication because most of the toxicity prediction to date is based on something known as QSAR technique, or which is traditionally known as quantitative structure activity relationship, which basically is based on the premise that if you have two chemicals which are similar, then activity or target space will be similar. So you cannot actually always rely on this principle to predict chemicals which are likely to become similar. So this is where we ended our first paper, posing a challenge, and suggesting that there are much more detailed models incorporating other insights which are needed to predict disease problem. I should mention that I showed low correlation, but there are so many toxicity predictions which are out there. These are computational tools. So here we wanted to test also that is our inference correct or aligned with what we see. So in our database, we have 157 chemicals which cause hepatotoxicity, and 185 chemicals which cause cancers. Okay. So there are these various tools. These are very highly cited tools which are used to predict today hepatotoxicity and carcinogenicity, and we actually computed the sensitivity. And as you can see, they are really low for the existing tools. One tool actually doesn't predict any of the carcinogens. Okay. So in general, these computational tools require improvements. Okay. So to summarize, what we did this period is that we of course identified EDCs with published experimental evidence. We made a resource by we classified them based on supporting evidence. We classified them based on environmental sources. We classified based on chemical properties. We have all this information into a resource which you can assess, and then we have done analysis which are network centric, the chemical similarity network, the target similarity network, the lack of correlation between chemical structure and target genes. We have also compared with different lists which are regulations or guidelines available out there. So in subsequent work where we updated DEDuCT again, we asked, I'm going to change, and we asked a different type of question. So the question we asked is that all of this work we were doing in actually purely in academia, blue sky research. Can this research on EDCs guide chemical regulation? And I'll summarize with 2 slides this lot of work over done over 2 years. What we did is that we compiled in a diligent way 36 chemical regulatory lists. These were regulations in different countries or inventories of chemicals which are used in different things like food contact materials and so on, and also some guidelines. And we compiled EDCs not only in our resource but other reports as well. So what I'd like to give a 1 slide summary of what we actually showed in this one. First, we show that if you look at the published literature, which is captured by PubMed, there is actually growing research efforts on endocrine disruptors because the number of publications on which containing the evidence over the years is increasing. Second, we compiled potential EDCs from various resources, and then we basically compiled regulations, 36 chemical regulations, guidelines, and inventories, which can be further classified into things like plant protection products like pesticides, things which are leach chemicals, things which are in water quality, and so on. Okay. And then what we try to see is that how often are these chemicals regulated in this list or indicate usage in different categories. So what we find is that these EDCs are both regulated and also in use. And what is even more disturbing is that some of the EDCs in particular, which are in use, are actually amongst the list of most highly produced chemicals in the world. Okay. By comparing against the list of highly produced chemicals, which is published by European Union and US. Okay. That's the summary of it. Okay. So what this means is that this kind of suggested that if regulatory bodies take into account information in our academic research, they can regulate better the chemicals which are potentially toxic and in use in our day. So I'm just going to give quick summaries of 2 works. So I mean these are all works which took 2 years actually each. So now the question of mode of action, right? So the OECD basically has come up with a framework to organize the information published on toxicity associated with the chemical. And the framework is what is known as an adverse outcome pathway. I know it's a bit of a detail. So an adverse outcome pathway or AOP is a model that identifies the sequence of molecular and cellular events required to produce a toxic event when an organism is exposed to a substance. And this framework allows the organization of the existing knowledge or toxicological information into different levels of biological organization. So there are, this is very detailed, and so there is a repository which has been created, and they are actually organizing all the knowledge into AOPs, and these AOPs will then be used to develop testing strategies for chemicals. And this is of course collaborative. So what we wanted to do, and which had not been done, is to carve out from the list of AOPs which the community effort has created, a AOP-wide network specific to endocrine relevant disruptions. And this has not been done before our work for the first time. So I won't go into the detail. Through a very thorough workflow, we actually created the first endocrine relevant AOP network, covers 48 AOPs in their AOP wiki and so on. And this is the first of its kind work in endocrine disruption, and this is also the first work in India on AOPs. And if you zoom into these nodes, which are actually a AOP ingredient, you actually see various molecular initiating events, key events, adverse outcomes, and so on. What is nice about this framework is that this is not only at the molecular level, but it's also at the organism level and the population level. It tries to capture all the aspects. So this we have taken a graph theoretic approach to it. So what we can actually learn from these AOP networks are points of convergence of different adverse effects, points of divergence from one system to another, that is from reproductive system to say developmental system. How the events are linked between different chemical stresses, that is between releases. And of course the further analysis of this network topology will provide insights on the underlying biological associations among endocrine disruptors, which chemicals are similar disruptive properties. And of course, this can help in better risk assessment of diseases. Lastly, to the question I think you were hinting at. So we mentioned one challenge in predicting disease, that is, the chemical similarity is not highly correlated with the target similarity. So we wanted to dig deep better and try to identify some other potential challenges in predicting disease. So as I mentioned, this framework of quantitative structure activity relationship of QSAR predicts inaccurate results because the basic premise may not be true, that is similar chemical structure may not be equal to similar activity. And in particular, there is a lot of interest which has been generated in identifying pairs or group of chemicals that are similar in structure but significantly differ in activity, known as activity cliffs. So you can think of this in some high dimensional space where you have chemicals which are similar next to each other but differ in their activity, the high peak activity. So you want to identify these cliffs. So we wanted to show that this concept of activity cliffs actually is a challenge, in particular in identifying diseases as well. And for that, we were able to identify a data set in published literature where the same group had characterized the activity of 202 chemicals, which are natural, synthetic, or environmental chemicals, in a single controlled assay. And so they had characterized this activity with respect to one of the prominent endocrine receptors, which is the receptor which is the androgen receptor. Androgen is important hormone in males. And again, I won't go into detail. This is again 2 years of work with a lot of methods. So we use this approach called structure activity similarity map to identify activity cliffs within this data set. So for example, this SAS map is very easy. There are other tools also we use. You have on the x-axis similarity of chemical structure. So if you have a higher number, each dot or each dot is actually a pair of chemical. So this is a pair of chemical which is highly similar, but their difference in activity is also low. So if you are in this third quadrant, you are very similar but very different in activity. So those are the pairs of chemicals which are the activity cliffs. So with this approach, we could identify many activity cliff pairs. Two have been shown there. As you can see, these are very, very similar chemicals, and this activity difference is in log scale. So it's 3 orders of difference in activity. So and this is actually the first systematic work of identifying activity cliffs associated with endocrine receptors. So with that, what I want to say that these are the challenges that we have found. Now, if we can identify these activity cliffs, how do we use it in our predictive models? Or how do we get out of this problem in predicting better is something one has to think hard. I have presented so far only challenges, but I would like to come back and tell you some solutions maybe in a year or so. So with that, I would like to mention that, of course, all of this work was done as a blue sky research. What was nice is that when we published in 2019 DEDuCT for the first time, it got both national and international attention. You see the C and E covering. This is Hindustan Times covering. This is the European Creative Institute, which is a sort of a body which protects the rights of workers across Europe. They cover in Europe. All this is great, but what was nice is that could academia or research in academia really influence policy making? So what I want to mention is that the ANSES, which is a French acronym, which in English translates to a government body, which is French Agency for Food, Environmental and Occupational Health and Safety, which is actually interested with coming up with this sort of policy for France, which is SNPE2, which will regulate all endocrine disruptors in complete France. They actually released this report in 2021. This is a 100-page report, 13 French and 17 English, which is an open thing, which is submitted to the French Parliament. They evaluated 27 worldwide initiatives on EDCs resources, and they, after their thorough evaluation over 70 pages, came to the conclusion that DEDuCT is the one they want to go to, and they actually used their complete list of 686 chemicals in our first list in their prioritization procedure and identified 16 chemicals which they have recommended to be sort of banned in coming years from now. So there are some of the excerpts of the report which I highlight. This is a report which is there. I am very happy because rarely you do something and somebody really uses it. So is that I'll try to summarize what we are able to do in this summary figure from my students' thesis. So we have compiled and curated diverse group of environmental chemicals. This is a lot of manual curation work, and which is based on published experimental evidence. So these resources that we have created are knowledge bases of environmental chemicals which are potentially toxic. Then we are able to link the exposome and health using network science. That is, we are finding perturbed target genes, adverse outcome pathways, exposure disease associations. I could show that. We have also done a study on that. How similar exposures could lead to similar disease outcomes by computation approaches. We have characterized the environmental chemical spaces using approaches such as chemical similarity network. We have also analyzed the physicochemical properties. For example, I couldn't show that, but we show that the chemicals which have the higher propensity of going from the plasma to the breast milk have a certain type of physical properties which the chemicals which would have less propensity do not have. Okay, that's something we have shown. Finally, we can actually use all of these also for regulatory assessment. For example, by comparing with chemical lists that are part of regulations, guidelines, or inventories. And finally, we can, bodies could use this information to prioritize chemicals of concern. With that, I'll just thank all the people who did the work. So the people who are in bold, actually, they contributed. Most of the people are gone. The person who led it is actually Janani and B.S. Karthik. And Janani did most of the work. She is now working in Novel Office as a research scientist. And these are my other collaborators. So thank you very much for your attention. And we take questions. And if there are still students in Krea, I will be happy to have them if they would like to do project positions, PhD, or if there are postdoc positions in university. Thank you for your attention and really thank you. Sorry I went over time actually.